This is yet another screen recording, this time to help you with eCRISP and mapping the guide RNAs back onto your diagrams. So you all have your diagrams of this coding sequence, the genomic DNA and the protein. Okay, so what we're going to do is use eCRISP to design guide RNAs, and then we're going to use BLAST in order to place the guides back onto our CDS region. So let me go over here and pick up my sequence. So I want my coding sequence, and I just need the gene name, okay, for eCRISP. So we're going to select Arabidopsis, and we found out today that you have to be all capital letters, so capital A, capital T, 5G, and then 16590, okay. And today we want to go to strict and select guides. If you get weird errors, check that everything is capital, and sometimes just rerunning the eCRISP will clear up those errors. Okay, their servers kind of funky sometimes. Now, what we're going to do, here's your output. Go down here and you can see here the guides that have been chosen. Okay, and here is the genomic DNA. Here's the region of the genomic DNA that it used. Here's the cDNA, or the full-length cDNA, and here's the CDS. Okay, and it's showing us the location of these introns. So we need to pick guide RNAs that surround our kinase, and surround the transmembrane domain. And I'll show you how to do the transmembrane domain, and then the kinase you'll be able to do on your own. So here are the guides, but the problem is that if we go up here, so here's a guide that might be near my transmembrane domain, so 1_493, 1_493. Unfortunately, this one matches 3 different genes, so let's ignore this one for now. Right now we want to find the ones that just hit one gene, and that is our gene, this one right here. Okay, so let's see right, so we're just going to be interested in these that have 1. Okay, and the problem is let's click on this. So what shows us a location, so 2641 to 2684 is the location of this sequence, and there's a guide here that it seems to be a good match, and so is recommending this as a guide. The problem is this number 2641 is not a useful piece of information because 0 is right here. Okay, and so this is somewhere over here, but we don't know where it is relative to our numbers. Okay, so this is 0. Our numbers start at 1 right here, right on the full-length cDNA. So, and then we have another coordinate starting here at 1, so that so we have 3 different coordinate systems, and they have yet another coordinate system. Okay, so to make it a little simpler and not have to do a whole bunch of problem solving, what we're going to do is use BLAST. Okay, and that'll also be good because if you notice, my gene is pointing to the right, which means it's on the top strand, and all the numbers are going to make sense. Some of your genes are on the bottom strand, and what you find here is going to be in reverse, so that the beginning of your gene is going to have higher numbers than at the end of your gene. Okay, and so what we're going to do though is use BLAST to show the location relative to the coding sequence. Okay, so the first thing we need to do then is go way up at the top again and collect on the Excel formatted tabular report. Okay, so this will give us a spreadsheet, and if you don't have Excel, you can open this in Google Docs as well. Now here we go, here's all the guides that it picked, and it's going to have all the ones. We want to make sure that it's only that has 1 match to the genome. Okay, so let's go and Okay, so here we go, number of hits. So we want to just start here. Okay, here and up. So what we're going to do, and this I'm going to take a couple of seconds to do, is we're going to build a little FASTA file. Okay, so we're going to build a FASTA file with this as the number, and this is the number, as the name, this is the name, and this is the sequence. There. Okay, so I'm going to open Word would be fine with this. You could do it in your e-notebook if you want. So what we're going to do is we're going to do angle bracket, copy, paste, okay, and then copy the sequence of that, copy the sequence, and then remove the NGG, okay, like that. Okay, do this a few more times. Actually, this might speed things up. Okay, so let me show you something to try. So let's go to Find, Edit, Find, Replace. Okay, we're going to replace AT tab with this AT. That's not going to work. So we're going to do AT5, replace it with AT5. Oops. AT5, and replace it with this. Okay, there we go. Oh, and one other thing. Let's see, can we do this? Nope. Oh, that's not what I want to do anyway. Oh, okay. Forget that. But then at least we've done that, so we can go and put a return in. Okay, now that's a mistake. Fix that. And now we just copy the sequence going down the list. Actually, I'm going to copy all but the first one. Keep text. And then just put it up here. Like that. Cut and paste. Cut and paste. Sometimes you just have to do the long way to get it in order to get the format that we need. And you may not need all of them. You can probably select 3 or 4, around 2 to 3 around each one. I'm just doing it this way to demonstrate what it is, what we can do. Okay, so there's my little file, and probably should save that or copy and paste this now into my notebook in case we need to repeat it, but we have what we need. Okay, let's put this away for now. So what I'm going to do is go to Blast, Nucleotide Blast, and click this box, Align 2 or more sequences. Okay, so it takes away the choice of database because we're going to tell it what sequence to use. So we've got our mini little database. So this is our mini database. Copy that and paste it here. Okay, so there's our little mini database. And then I'm going to go over here and get my CDS sequence like that. Okay, so that's my sequence I'm going to compare to. and that goes in the query box. So the query box is our CDS. Okay, and this is our mini database, except that for some reason that sequence didn't come along. There. Okay, now click somewhat similar sequences. Okay, critical step. So we've got our CDS in the query. We have our mini database as the subject, and then we're going to click somewhat similar sequences, blast in. Okay, this is because these are short sequences, and for it to work, we need to do it like that. And there we go. There's our alignment, our output. So in order to so what we're going to do is go back here over here to alignments, and there we go. There's our numbers that are relative to the cDNA. So I want something that's around 700 to 798. So I look through here, so that's kind of close. Okay, 100, that's not too close here. This one's, oh, here we go, look at this one. So 582 to 601. That's really close to my transmembrane domain here. So let's go back now and see. This is AT5G underscore 1 underscore 986. So let's go find that one. Underscore 1 underscore 986. There it is. Okay, it's nice. It only matches to the gene, the match string info. It's a perfect match. There's one mismatch. That's okay. We'll go with it. Okay. It has the NGG right there, which we want to see, and the SAE score is really high. And like we talked about in class, the S is how specific it is. This annotation means that it's in an exon, it's not in an intron. And E is some sort of efficiency. How well do they think this primer will work? So those bars are all nice and long. So that's what we want to see. So then I would copy and paste this into my E notebook. Actually, so this is one potential one. Okay, so I would have so upstream would be that. Okay, and then go over here and screenshot it. Okay, and I'll upload that in a few minutes to my data. And let's go ahead and screenshot this. There. And highlight it. And actually what I can do now, because the Mac makes it really easy, is if I can find it, 596, there it is. So that's the gene I want. So I should be able to edit that. And let's see if I can put a circle around it. There we go. Put a circle around it. So I'm going to use this one. Also, you would like to go back over here. Oh, that's done. Go back over here and let's screenshot this because it has the numbers that we want, 582 to 601. So let me put that in here. .601. And so then later I will go in here and draw in I'll draw in this guide here and call it my guide RNA. I'll give it a color and do a color over here, and then I repeat for the downstream one for this, and then I repeat for the kinase. Now if the kinase domain for some of you some of you, the kinase domain is actually like this over an intron. That's okay. Just make sure that no guides are on top of this junction, right? So we don't want a guide like that. So this guide for the downstream, if I can't find one that's right there, then I'm going to go over here. But make sure it's on one side or the other of the exon-exon junction. Okay, we do not want it to overlap because that doesn't exist in the genome, right? It's interrupted by this intron. So we need the guide either here or here, here or here. In some cases, it might have to overlap into the transmembrane domain or into the kinase domain. That's okay. It's just whatever guides you have to work with. And we'll talk about in class different scenarios that you come across. So there you have it. That's how you can go from the eCRISP coordinate system to our coordinate system using BLAST. Okay, so good luck, and we will talk more in class.